Well, good evening, everyone. Actually, let me just check this one thing here. This is partly a review and partly some new information, since I presume that not everyone on the call tonight heard my talk in 2019 in Baltimore. So I thought I would first run through some of the basics just to bring everyone up to speed on what KAT6A is and what it does or is thought to do. We all know that KAT6A is associated with developmental and physical abnormalities, and I've been focusing on the metabolic abnormalities that are related to the deficiency of KAT6A. And one of the reasons to focus on the metabolism is because because KAT6A disturbs metabolism, there are many ways to put metabolism back on track. And as long as you understand what is causing the abnormalities, then you can try various therapies to get things working better. So I'm going to go through again what the role of KAT6A is in gene expression, that is how to turn genes on and turn genes off, in which KAT6A has a very important and widespread role. KAT6A is a so-called acetylation enzyme. It adds an acetic acid group, vinegar basically, an acetic acid group to histones, and histones are the protein that coats DNA, and histone plus DNA equals chromatin. So KAT6A is a protein that interacts with the histone chromatin complex and causes changes, so-called epigenetic changes in the DNA, or there are epigenetic changes in the DNA which produce sites that are favored by certain of these histone modifiers, and the histones themselves are modified in a way that makes them bind to the DNA and therefore shut it down, or open up the DNA and allow the enzymes that can convert the instructions in DNA into RNA and then into protein. It allows those to interact with the DNA. And this is a very dynamic process in all cells, that the DNA and the histones are constantly being modified based on the needs of the cell. As you know, all cells have all 20 some odd thousand of our genes, but they don't all work in all cells, and they don't work all the time in the cells where they're supposed to work. So this is a process that interacts with the needs of the body. Specifically, when looking at acetylation, the adding of the acetic acid group to histones, that is very much tied into diet, because the acetic acid groups come from diet. Then I'll show how we have found that citrate increases histone acetylation, and citrate we had found also improves gut motility, and I'll show why that occurs. Then we'll move on to newer topics, and I touched on this in Baltimore. That is, there are other ways to modify histones, or KAT6A can use other compounds besides acetic acid or acetate to modify histones, and one of those is propionate, which is a is this marker seen by everyone? They see my cursor?
Yes.
Okay.
Yes, we can see.
So propionate is 3-carbon. It is a 3-carbon unit. Acetate is a 2-carbon unit. So it's very similar to acetate. And propionate is used by KAT6A to modify histones. And that is not as well understood. That is, what parts get the same portion of the histone can be modified by acetate or propionate and other compounds. And so it's just understanding acetylation is hard enough, but we need to look into other aspects, especially propionylation, because that supposedly propionylation has more of a regulatory role. I mean, acetate has a regulatory role, but certain sort of major regulators in the genome require propionylation to open them up. And then we'll talk about triheptanone, which is a way of generating propionate, and then some other aspects of histone modification. So just to review the biochemistry and what is going on. So here is a complicated diagram, but these are the histones which are wrapped around the DNA, and they generally, when are bound to the DNA in circles like this or in loops like this, the DNA is shut down. And whether or not the a modification or to open this up, certain modifications have to occur, which are shown here. When the DNA is turned off, it's called heterochromatin, and that relates to what was seen under the microscope many years ago. And euchromatin is a chromatin that is working. U means good. So euchromatin is where there has been interaction, and I'll show in the next slide what is going on with acetate, that's that, and a methyl group, and the histones are acetylated or methylated. The DNA itself also is methylated, and all of these changes in the DNA and histone provide signals to open up the DNA to allow the RNA polymerase to generate an mRNA, and without generating an mRNA, the DNA can do nothing. So this is the major step in turning a gene on. To turn a gene on means that RNA is being made. And this just shows some of the other, there are hundreds of proteins that interact with DNA and chromatin. This is where KAT6A is. So KAT6A and KAT6B can be alternately incorporated into what's called the MOZ or the MOZ complex. And this is a complex of proteins that are major, that are very important in the regulation of the modifications that I just talked about. And the MOZ complex also is also quite complex. And this just shows that the basic substructure of the MOZ complex involves KAT6A and these other proteins. And as you can imagine, defects in proteins like BRPF also cause syndromes not unlike KAT6A. But anyway, there are these multiple enzymes or proteins that are also bound to the complex. And one of the more important ones is called HAT, which stands for the histone acetyltransferase. And this is what adds the acetyl group to the histone. And it specifically interacts with the H3 histone and other compounds or other enzymes or other proteins interact with other histones. And so it's, when I say it's complex, it's that there are variations on the theme for all of these different protein combinations, and therefore, and each one is adapted to turn on or turn off certain sets of genes in the cell. And it's still it's becoming better understood, but simply asking the question, what genes does KAT6A have any relevance to? It's not the easiest question to answer. I'm sure Dr. Voss would testify, and that's part of the work that she is doing. If we understood all the genes that are significantly affected by a deficiency of KAT6A, then we might be able, some we can figure out from the biochemistry. Others we have to look at what is going on, what genes are turned on or turned off if KAT6A is not there or only half of the activity is there. And generating that list of genes will also be important in trying to work out different therapies for KAT6A children and adults. This just shows another way of looking at this interaction. So here's the MOZ complex, and it shows that if there is methylation of this specific point, this is the fourth lysine molecule, or fourth lysine amino acid in the H3 protein. And when that's methylated, MOZ can't bind, it can't acetylate. And in contrast, when H3K9, the ninth histone in H3, is acetylated, and when H3K9 is methylated, the MOZ complex can bind. And what is, so there are certain changes, the acetylation of 14 of H3 and of K9, that's called a higher condition that it recruits, or the MOZ complex can interact and do its work in gene expression. And fire is used to say that these modifications of the H4 and H3 histone block the binding of MOZ. So that's another level. There are other proteins that cause the substitution, so that's another level of regulation. So where were we several years ago in trying to understand if there is a metabolic abnormality in KAT6A? We actually had a strong clue, so to speak, long before Dr. Esper got in touch with me, and that is I had been called by a geneticist in Washington about a child who was found to have some mitochondrial type abnormalities, and I suggested a particular mitochondrial cocktail, combination of mitochondrial antioxidants and other factors, which we generally use when there's mitochondrial dysfunction. And that caused a significant improvement, which I wasn't surprised about given the biochemistry. But then several years later, that child was found to have KAT6A. So then Dr. Esper contacted me saying, well, let's look at this. Maybe if it worked for Chloe, maybe it'll work for all children with KAT6A. So I began to look at the biochemical abnormality, look more closely at the biochemistry of KAT6A, mostly looking at blood studies, amino acids and organic acids. And this is what we found was, there are certain characteristics we found, which I'm not going to go over here, which is a high level of two amino acids, asparagine and citrulline. And they're pretty, when you have both are increased, it's quite characteristic of KAT6A. And what we know is that, don't show it here quite, it's right around in the corner here. Asparagine is a very important compound that feeds into the citric acid cycle. And if that is high, there might be a problem in the citric acid cycle, i.e. asparagine can't get in. And when asparagine stays high, it produces this other amino acid called citrulline. So looking at this more closely, thinking about how one can acetylate. So here we are, the HAT protein, KAT6A with its HAT function, acetylating right here with DNA. What it uses to put the acetyl group on the HAT protein first, on the HAT enzyme first, and then that binds to, that is transferred to the histone here. So the question is, how do you increase the level of acetyl-CoA inside the nucleus where all the histones are? And this is the mechanism by which one can do it. Is acetyl-CoA is naturally made from citrate, which is part of what's called the citric acid cycle, or the tricarboxylic acid cycle. And that acetyl-CoA in the cytoplasm, in the mitochondrial space, gets out as acetylcarnitine. It also can get out, this citrate conversion to acetyl-CoA can also occur outside. But just showing a diagram here, giving carnitine enhances the transfer of acetylcarnitine into from out of the mitochondria into the cytoplasmic space, so between the mitochondria and the nucleus. And then it can go through a couple more steps to generate acetate, again, the acetic acid or vinegar, and that can make acetyl-CoA. Another effect of carnitine is carnitine in general increases the amount of CoA SH, which is called free CoA. It's coenzyme A that is able to bind to acid functions like acetate and to make a conjugate, an acylated CoA, acetyl-CoA. So carnitine was one of the major things that, or major effects that I suspect that carnitine was one of the major causes of the effect in Chloe, and that is because the mitochondrial cocktail has several parts, some of which work fairly quickly, carnitine and pantothenic acid, and some of which take 3 months to reach maximal effect. And the fact that we had now seen many children with KAT6A get better within a few days of starting carnitine suggests that carnitine is acting in this way, or interacting inside the nucleus to also work to make acetyl-CoA. So carnitine boosts the system somehow, and naturally we expect it could be acetylating protein, but it's going to take an awful lot to prove that that's what's happening. Carnitine itself can benefit children with mitochondrial problems for a variety of reasons, and therefore it might be that carnitine, though it could increase acetylation, is really simply making mitochondria stronger. And when I say mitochondria, this is the real business of mitochondria. Mitochondria make ATP, make energy, and that occurs in the electron transport chain. That's a very simple or straightforward process of taking an electron and turning it into energy. But to generate what feeds the electron transport chain, one has to have a running citric acid cycle. Here's the asparagine here, and this is a very important point in the citric acid cycle for controlling metabolism. And you might think that if that asparagine might be high because it's not being converted into oxaloacetate, which might cause a deficiency of citrate. And even if citrate is not being made in adequate amounts, giving carnitine can help. But this was a suspect. Maybe asparagine is high and citrulline is high because there is an inadequate level of citrate. And sure enough, all but one child that I've tested with KAT6A has a below normal level of citrate. And after treatment with citrate supplementation, which I'll go into later, that's what they get better. So there's a the presumptive problem. Again, this is secondary to the regulatory abnormality caused by KAT6A. The presumptive, one of the metabolic consequences is deficiency of citrate, and therefore a deficiency of acetyl-CoA. This citrate is divided into acetyl-CoA and back into oxaloacetate. And this is an important compound which we'll come to later, acetylcholine. Any acetyl-CoA that's not utilized is generally turned into fat. There also is a mechanism that carnitine might help, because to make acetyl-CoA, there are two ways of making acetyl-CoA in the nucleus. One is to import acetate to bind with free CoA, and the other one is to convert another compound that comes mostly from glucose, pyruvate. Pyruvate is essentially a glucose molecule split in half. Pyruvate can be converted into acetyl-CoA directly within the nucleus. This is only recently found that there is this enzyme which is better known outside the nucleus, but this pyruvate dehydrogenase also exists in the nucleus, and it appears to be the same pyruvate dehydrogenase that we know can be stimulated by carnitine. So this is another mechanism by which giving giving the mitochondrial cocktail or just carnitine and pantothenic acid by itself may work. Pantothenic acid, just to mention, is the vitamin that is used to make coenzyme A. So we often include pantothenate, vitamin B5, in a mitochondrial cocktail to increase the level of coenzyme A, which, as you can say, if we can increase that, then we can increase the production of acetyl-CoA wherever acetate can go. How that is important with regard to some of the problems of KAT6A, I had figured out many years ago that children who have severe dysmotility, what's called in mitochondrial diseases, is called pseudo-obstruction. When it's really bad, the gut behaves as if it's got a block, but it's really just a functional block, the so-called pseudo-obstruction. But that does occur in KAT6A, but more common are these episodes, sometime almost every day, sometimes periodic, of extreme colicky pain, as it's called medically. It's a kind of severe stomach cramping that is very, very painful and obviously distressing to children, parents alike. And I'd found that I had reasoned that, a deficiency of acetylcholine, which is the major neurotransmitter that makes gut muscle contract. So you have to be able to have enough acetylcholine, which requires, which is made from acetyl-CoA, which is made from citrate. So citrate to acetyl-CoA to acetylcholine. If acetyl-CoA is low, then acetylcholine is going to be low, and the gut can stop working. And it is it's pretty dramatic when it's used in classical pseudo-obstruction, that someone who is not able to stool except with with strong stimulants every 3 or 4 days can start stooling 2 or 3 times a day, again within about 24 hours of starting citrate. And citrate can be given orally as tricitrates, or one can make up almost the same amount that is needed to stimulate acetylcholine synthesis by drinking a lot of lemon juice. And as I mentioned, this kind of dysmotility occurs in children with other types of mitochondrial problems. And I mentioned in 2019 that one of the boys with autism that I had treated for a mitochondrial problem, his parents told me that they had to lock the freezer, put something to keep the freezer, the refrigerator closed, because their son would go in and drink down a whole bottle of real lemon juice. And that really, that's essentially the same as giving tricitrate. So he had figured out himself that he needed citrate. So to save the real lemon, I wrote a prescription. So this is where we were a while ago. And this summarizes mitochondrial treatment that some children, this is not as important in KAT6A, but some children have trouble fasting, that is overnight, for 10 hours or so. And some children will then, we give them cornstarch, which helps extend their glucose synthesis overnight. Complex 1 is a part of the electron transport chain, which can be directly stimulated by giving carnitine and pantothenate. About 1/4 to 1/3 of complex 1 at any one time is not turned on inside the mitochondria. And by giving carnitine and pantothenate, a good deal of that residual activity can be turned on. One of the things that we see in older children with KAT6A is that the apparent mitochondrial dysfunction that we see by the various tests that we do can damage, can produce what are called free radicals, and those free radicals damage the mitochondria in an irreversible way until the mitochondria can can double and replace their membranes. It causes a long-standing damage. And by providing a specific combination of antioxidants, we can stop that process in most mitochondrial disorders. And therefore, and I can, when I do my various tests of children with KAT6A, I can tell by a variety of tests whether there is ongoing oxidative damage. And for those children who have oxidative damage, we give a more complete mitochondrial cocktail. Another thing that happens in mitochondrial disorders, not very commonly, but it has occurred in several children with KAT6A, is that if you, let me go back for a second, if citrate is not being made and you can't generate this side of the citric acid cycle, amino acids are taken and turned into what are called citric acid cycle intermediates. And one of those is an amino acid methionine. And because methionine is used for many different processes, and it's also not especially abundant in mitochondria or in protein and in the diet, it will become deficient. And some of the tests that I do are looking for that deficiency. And when I find that, then we give them methionine. Sometimes other amino acids are deficient, like arginine or tyrosine. And then I'd worked out a long time ago what are the optimal doses, which are given here. And then citrate supplementation, it's used for treating kidney problems and severe mitochondrial problems, and often one requires 5 milliequivalents per kilo per day. But we find that in KAT6A, it's not a huge amount, it's between 2 and 3. So I usually start out a child at 2 and work up in steps to 3. And there's usually a transition point over a narrow range when the gut starts to work. So the conclusions at the end, in 2018, when I was doing most of the initial biochemical work, was that the children, KAT6A children, have signs of mitochondrial dysfunction, and that treatment of the children leads to verbal and motor improvement, reduces the severity of infections according to some parents, and now we know that it also decreases complications like the dysmotility shown here with citrate or methionine. There's one boy who was getting transfusions of red cells because his bone marrow was essentially turned off, or his red cell production was turned off. And one of the characteristics when methionine is deficient is the red cells are large, so-called macrocytic, which people suspect is caused by B12 deficiency, but it's actually in mitochondrial problems usually due to methionine deficiency. So we simply give methionine and the problems go away. So to move on to topics that we are working on or hoping to address in terms of understanding or in terms of working on better treatments or additional treatments for KAT6A. So we know that, I should say in general that KAT6A here, when the children with KAT6A deficiency have only half the normal amount of this enzyme as part of the KAT6A complex, the histone acetyltransferase. So half of the enzyme is there, and oftentimes when only half of the enzyme is present, if there's a way to sort of boost the enzyme to push up the activity of the enzyme, it can compensate for many of the deficiencies. And that's a standard approach to treatment in metabolic disorders. So acetylation, it can be augmented by citrate and by carnitine, but histones are also modified by other groups, as shown here. Methyl groups I had mentioned earlier, that's just a carbon with 3 hydrogens, and that is mostly used for methylation of DNA, or it's much DNA is turned off by methylation, but also histones can be methylated, the lysine of histones. Acetyl group, the propionate groups I had mentioned, but also butyryl, which is 4 carbons, and crotonil, which is 5 carbons with one unsaturated bond. And they all have, this is just showing a schematic of the different histones. There are 5 main histones. Actually, there are several types of H1, but there are 5 principal histones, and they each have their own particular pattern of histone modifications. So we know that these groups here, the acetyl, propionyl, butyryl, and crotonil, all require KAT6A or KAT6B for these to be used to modify histone. So we know that we're helping by augmenting acetylation, although that has not been specifically proven yet in a child, a person. But we need to understand, so we would expect that these other modifications will also be deficient. So now that we seem to be able to modify acetylation fairly well and compensate for some of the metabolic deficiencies that occur, we also want to focus on what these other, how much KAT6A contributes to these other modifications and what to do about it. For example, giving lots of acetate or in the form of citrate. So again, the citrate converts into acetate. By giving lots of acetate, we can change gene function. But since they're all deficient, how do we increase, if propionylation is deficient, just by increasing the amount of propionate or increasing the amount of butyrate, it's not as simple as that, because what histone, which position in a histone is modified, is not determined as much by the level of this compound as by other signaling factors. We know that acetylation is deficient, so we can augment that. In theory, we could do the same thing by giving acetate, propionate, and butyrate. I wouldn't give this, it really stinks. But it may not, by just throwing in propionate, which we can do fairly easily, that might help and it might not. So we need to try to understand more about what regulates this aspect of KAT6A function and whether or not we should, and how much of certain other acylation compounds we should give. And the one to focus on first is propionate, because it's been shown that some of the histones that bind to master regulators, let's call them, in chromatin, preferentially bind propionate. And there the same HAT enzyme is responding to signals on the chromatin in terms of whether it puts on an acetate group or a propionate group. And just by throwing a lot of propionate and acetate into the mix, it might not work, or it might even cause trouble. So that's why we haven't leapt ahead with giving propionate. This is just to show the complexity. This is a histone H4, just one of the, actually it's mostly H3 and H4 that are acylated. It just shows that of the different compounds that are substituted on different sites in the protein. So these are all the potassiums shown here, and acetate is right there. So potassium 44 in H4. So this again is another way of looking at how complex the system is, and what we need to do is find the right balance between stimulating acetylation, which is the easiest thing to do based on the compounds that are available to us, but to understand whether augmenting the propionylation, which is equal with acetylation at this site, it's hard to know a priori whether that's going to help or hurt. These studies are underway, and as you see, the illustrious Dr. Esper is an author on this recent paper that shows that indeed propionylation in what they did, they tested cells from patients and from cancer cells and showed that indeed propionylation of histone H3 is deficient in the cells of patients with KAT6A. So that's a proof of principle or proof of the deficiency of propionylation. And therefore, it's a way of saying that we do indeed need to focus on propionylation. The other aspect of treatment that, or the other way to generate all these short-chain fatty acids, acetate, propionate, butyrate, is from gut metabolism, from gut bacteria metabolism. And we all hear that it's important to have fiber in the diet. And many years ago, I thought, well, that's bulk to give fiber to the as part of the diet. It helps with the stool bulk. But no, it may do that, but one of the major roles of fiber is it serves as a food for gut bacteria. And one of the major byproducts of gut bacteria when they metabolize fiber is to generate short-chain fatty acids. And all these groups that I've talked about, acetate, butyrate, propionate, those are short-chain fatty acids. So the gut bacteria generate the short-chain fatty acids, and then these are then metabolized in the, they're either passed through the cell, this is the intestinal gut wall cell, is either passed through the cell as free short-chain fatty acids into the liver, or it can go, it can be metabolized within these cells and just broken down into CO2. So there's a lot of studies on the microbiome, as it is called these days, and various claims, documented and undocumented, that taking probiotics and other compounds that stimulate or alter the gut bacteria has some important physiological or developmental effect. It's been studied for years in treatment of autism. And there probably is something going on there, but again, the system is so complex that it's hard to know what to do with the how to modify the gut bacteria. Let's say we felt that propionate was very important, and we wanted to increase the production of propionate in a natural way. It could, in theory, be possible to get bacteria that generate propionate more than others. There is a genus of bacteria called Propionibacterium, so that might be possible. But everyone's microbiome is different, and diet affects the microbiome. So this is another level of complexity. And for now, we are trying to work out treatment methods like citrate or giving propionate in a medication form. But this is not unimportant. It's important to think about the natural production of short-chain fatty acids, because after all, the propionate, we can make large amounts of propionate by giving a medication called Trihephenoline, but obviously long before Trihephenoline came around, there were natural ways of doing it. So that's another area of investigation. And just to mention, Trihephenoline is it's like MCT oil, if some of you are familiar with that. That's a 10-carbon fatty acid. This is the glycerol base, and then a 7-carbon fatty acid, 3 attached to the glycerol. And that heptanoic acid is broken down into 2 acetic acid groups and 1 propionate group. And the propionate this will raise the level of propionate for acylation by histone, but it also is being used in the treatment of certain metabolic disorders because it can generate extra energy through the citric acid cycle. Propionate that is not used directly for histone modification, which is the bulk of it, goes into a compound, it's converted into succinyl-CoA, and then that's metabolized simply to generate energy in the citric acid cycle. Fortunately, there, especially for what are called long-chain fatty acid oxidation disorders, triheptanone has been studied and is for many years, actually about 30 years, and it has undergone some rather intensive clinical trials in the past 5 years. So we can benefit from some of the experience with that in terms of side effects and in terms of how much of triheptanone has to be given to significantly increase propionate. The curious thing is when I looked at the metabolism of children who are on triheptanone, it turns out that propionate itself was not all that high, but the 5 carbon, so the propionate plus 1 acetate group, or pentanoic acid, was the highest compound. So yes, it generates propionate, but it also generates the 5 carbon acid, so it's going to be modifying other sites in histone. So we have to go very carefully, step by step, treating children with KAT6A with as many non-metabolic ways that we can, and trying to make some advances in finding additional metabolic treatments or to refine the metabolic treatments that we have. But at least it is one of the interesting things about this, and why I presented this information at the David Smith meeting, is that that's a meeting of dysmorphologists, mostly the doctors who see patients with conditions like KAT6A, and they don't generally think metabolically. So I wanted to show them that a child who people were very surprised that KAT6A had measurable metabolic abnormalities. And without Chloe having that diagnosis first, we might never have known. But I thought it was important to say that, to show others that they really need to look at their children with developmental problems, no matter what the cause of their genetic defect. So, so this is a summary of where we are with KAT6A treatment now. So again, the mitochondrial antioxidant, the mitochondrial activation and antioxidant treatment, methionine, and sometimes some other amino acids are needed, citrate, and then, as I mentioned, dietary fiber management, and triheptanone supplementation, which is what we're working on the most right now. So we're still continuing to gather more metabolic data on as many patients as we can. KAT6A mouse exists. Dr. Voss is intensely studying her KAT6A mice, and the various medications, ways of increasing acylation to be studied, obviously more safely in the mouse. But for certain, any candidate compound, we are hoping to have a trial. We hope to have it this summer, but we may probably start next year. And a tentative thought of, if all the preliminary data that I can generate look good, is to use triheptanone as part of that treatment protocol. I think that's it. So I don't know how I get out of this. There we go.
Thank you, Dr. Kelley. Thank you.